In today's video, we are going to focus entirely on DNA, which is one of the most important molecules in biology. We will walk through the structure of DNA, paying special attention to nucleotides and the concept of complementary base pairing. By the end of the explanation, we will also briefly explore how a gene can ultimately code for a protein, which plays a vital role in almost every cellular process. The first thing to understand about DNA is that it is composed of two strands. These strands are wrapped around one another to form a spiral staircase-like shape known as a double helix. This twisting double structure is iconic and essential to its stability and function. In order to understand the rest of the DNA structure more clearly, let us imagine untwisting just one of these strands and laying it out flat like a ladder shown as a two-dimensional diagram. Now, when we take a closer look at this single strand, we can observe that DNA is actually a polymer. That means it is a large molecule made up of repeating subunits. These repeating subunits are called monomers, and in the case of DNA, the monomers are known as nucleotides. Each nucleotide consists of three main components. At the top of each nucleotide is a phosphate group. This phosphate is chemically bonded to a sugar molecule, specifically a deoxyribose sugar, and on the side is one of four different nitrogenous bases. These bases are adenine, thymine, cytosine, and guanine, often abbreviated to the letters I, T, C, and G. These four bases are what differentiate one nucleotide from another. That means all nucleotides have the same phosphate and sugar groups, but the base is what makes each one unique. Therefore, there are effectively four types of nucleotides in DNA, each corresponding to one of the four bases. When two nucleotides are joined together, the phosphate group of one nucleotide binds to the sugar of the next nucleotide. This bonding process repeats again and again, thousands of times, to form one long chain. The repeating sugar and phosphate groups form what we call the sugar-phosphate backbone. This backbone acts like the outer rails of a ladder and helps protect the information-rich bases which stick out from the middle. When you look at the complete D and I double helix, these sugar-phosphate backbones make up the sides of the helix, while the bases point inward and pair up with bases from the opposite strand. This brings us to the very important idea of complementary base pairing. If we line up a second D and I strand running in the opposite direction, we will see that bases from each strand pair up with one another across the middle of the helix. But not all base pairs are allowed. Only certain bases can pair with each other. Adenine always pairs with thymine, and cytosine always pairs with guanine. This rule is known as complementary base pairing. Because of this, if we know the sequence of one D and I strand, we can accurately determine the sequence of the complementary strand. For example, if we had a sequence that read adenine, guanine, thymine, guanine, cytosine, thymine, thymine, adenine, cytosine, then the complementary strand would be thymine, cytosine, adenine, cytosine, guanine, adenine, adenine, thymine, guanine. Every base has its specific partner, and this pairing helps to keep the structure of DNA stable and allows for accurate copying during cell division. Now, when people refer to the genetic code, they are really talking about the specific sequence of these bases along a DNA strand. A gene is simply a particular segment of bases that contains the instructions for building a particular protein. To do this, each group of three bases is called a triplet, and each triplet corresponds to one specific amino acid. For example, the triplet adenine-guanine-thymine may code for one type of amino acid, while guanine-cytosine-thymine might code for a different one, and thymine-adenine-cytosine might correspond to yet another. Now, to see how this whole system works together to build proteins, let us take a longer sequence of DNA and look at how the cell uses it. First, the cell reads the DNA sequence three bases at a time, identifying each triplet. Then it looks up which amino acid each triplet codes for and assembles those amino acids in the exact order given by the sequence. These amino acids are linked together to form a long chain. Once the chain is complete, it folds into a specific three-dimensional shape to become a functioning protein. The final shape of the protein is determined by the exact sequence of amino acids, and that shape gives the protein its function. Proteins are involved in almost everything cells do. Some of the most important types of proteins are enzymes, which act as biological catalysts to speed up chemical reactions. Hormones are another type of protein, responsible for transmitting signals and messages around the body. And then there are structural proteins that give strength and support to our cells and tissues, forming key parts of skin, muscles, and even hair. So in summary, the structure of DNA is essential to its function. The double helix allows for stable storage of genetic information, complementary base pairing ensures accuracy in replication, and the sequence of bases forms the genetic code used to build all the proteins our bodies need. Understanding this process gives us powerful insights into biology, health, and how life itself works at the molecular level.